Good afternoon. This is Dr. Polaiah. I'm very happy to deliver today a video lecture that is on visual perception of color images. As we are all in the continuation of listening these video lectures as one of the topic which we are going to cover from our syllabus, that is from the image processing. So that topic name is, we can say boldly, that is visual perception of color images. So how your visual perception of color image you are looking based on the human visual system, that is HVS. A true measure of image processing quality is how well the image appears to the observer. HVS is very complex and is not understood well in a complete sense. However, many of its properties can be identified and used to our advantage. So lens is flexible and its refraction is controlled by the thickness. So thickness is controlled by tension of muscles. So the muscles which are connected to the HVS. So this is the meaning of HVS, how we are looking at an image. Coming to this HVS, we can extend in terms of where we can concentrate this digital image processing as a field is built on a foundation of mathematical and probability formulations. Human intuition and analysis play a central role in the choice of one technique versus another, and this choice often is made based on subjective and visual judgments. The major structure of the human eye, the eye is nearly a sphere with an average diameter of approximately 20 mm. 20 mm. Three membranes encloses the eye, the cornea and the sclera out of outer cover, and the choroid and the retina. So by using these components of the membranes with a diameter of 20 mm, we are going to be see as the structure of human eye. So here the visual perception of color image is also we can concentrate on how the HSV, how your human visual concentration of we are concentrating is here. This is innermost membranes of the eye is the retina, which lies the inside of the walls entire posterior portion. When the eye is properly focused light from an object outline, the eye is imaged on the retina. So the cones in eye, each eye number between 6 and 7 million. So they are located primarily in the central portion of the retina called as fovea, and are highly sensitive to the color. So this is color what we are concentrating in terms of humans can resolve the fine details. These cones largely because each one is connected to its own nerve end. The muscles controlling the eye rotate the eyeball until the image of an object of interest falls on the fovea. Cone vision called photopic or bright light vision. So based on this, your color image, your image of the color may going to be identified. So you are supposed to understand such a theory in terms of theoretical, how we can connect with the physical system. We can say that in terms of structure of eye only. So the structure of eye is right now we can say that center of retina which concentrating on towards to observe the color image. So how in this sense you can understand by seeing the structure of the eye, as in terms of lens, how it is connected, okay, and what's the important of here, that is choroid and retina, and also we are going to be concentrated on cornea. So based on this, we are concentrating how the color image is right now concentrate towards to be changes in terms of colors. That is, what are the colors? RGB color model. So that colors are which we are going to be concentrate based on the structure of IARE. That means R based, red based color we are identifying in the sense completely. It is changes of the observation of the human eye only. So if we change instead of red as a green, you can also understand that how that retina function will be concentrated. And similarly, blue also. So RGB play major role in the colors only. Okay. Next, such a thing which we are going to be keep on to the classifying of different types of images based on the structure of eye are number one binary, number two which we are concentrating structure of the eye are here in terms of three types of images as follows. Let me see out of three types of images. The first one is binary image. The meaning of binary is represent zeros and ones only. Zeros and ones. So that zeros, which we are zeros and ones, how can you keep into the in terms of image observation? In terms of image observation, which can be processed, it is the simplest type of image. It takes only two values. That is black and white. That is black and white, zero and white only. That means the binary image consists of one bit of the image. It takes only one binary digit. It represents a pixel. Binary images are mostly used for general shape or outline. For example, optical character recognition is the major visual perception of color images example. So coming to such a binary images are generated using threshold operation only, that is fixing information. That will fix when a pixel is above the threshold value, it is turned white. Which are below the threshold value, then they are turned back as zero. So after an image has been segmented into object background regions, then we may going to be understand one intends to represent and describe them in characteristic features for computer processing, and also during pattern recognition or in quantitative codes for efficient storage during the image compression process only. So coming to such a perception, we are going to be concentrate with certain example images which we call as structure examples. So structure examples as shown in below figure, which represent, you can compare how your binary image is right now, keeping in terms of colors also. How it is possible? Because your visual perception, as we are using HVS process, there we are concentrating how the colors images will be identified. You see here, there are two different color images based on the RGB model, which we are going to be used for the color image process. So what we want to concentrate is here, you are concentrating mostly on the aspect of the structure examples here. So how these structure examples are varying in terms of colors based on the perception only. So coming to that perception, if you wanted to understand in detail, let us to see that in terms of block diagram. So the block diagram is nothing but the functions of the blocks are going to be help us to understand how this HVS can be described in detail based on the structure of eye. We keep in terms of functions of the blocks. That means you can understand that one block of the output connected to the other blocks of the input. One block of the output. Output means this have input image, okay. This is the input image, okay, and that is optical process, which is we are seeing OCR, okay. Optical system is OCR, and process it. Then you are going to see this. This is called structure I, okay, and we are going to be keeping into the visual path. This is what we are seeing in terms of RGB colors. So you are going to have your color image in terms of your required output. So human eye is in terms of observation, how we are going to be concentrated is here in terms of observation is one by another. The looking at an image, how it is changes, and finding based on human eye, how we find out is one by another. It won't take too much time to help us to say that color image is in terms of red based or blue based or green based, how the color images of them happen. How can we understand in the sense one by another sequentially will be connected and getting the output in terms of your expected visual path. So coming to that, visual path is nothing but here rods in the eye are much larger than in terms of 75 to 150 million and are distributed over the retinal surface. The rods are not involved in the color vision and are sensitive to low levels of illumination. When the lens of eye focus an image outside the world into a light sensitive membrane in the back of eye called retina, the image is formed. The lens of the eye focuses light on the photoaspect cells of the retina, which detects the photons of the light and responds by producing the neural impulse. So this is the main information which happen at the internal part of the eye. So how let you see that? Let you take the one image. This is your image. Your eye is here. This is your structure of eye. Okay. So this graphical representation of the eye, looking at the palm tree point C is the optical center of the lens. Okay. So you are taking the 100 meters. Okay, lens, and the height is 15 mm. How this observation will be observed. Observation will be happen. So you are keeping the center is high. Point is keeping. This is your center point. We call as a point. This is point. See. So from this you are looking at your color. So you are changing these colors. Colors how it will be changes depends upon your observations only. So that's what we are concentrating here. Your visual perception of the color image is looking at from the center point C. The elements of visual perception is here also extend in terms of brightness, adaptation, and discrimination. That is, digital images are displayed as a discrete set of intensities. The highest ability to discriminate black and white at different intensity levels is an important consideration in presenting image processing result. That we can understand how this digital image processing result happen. Likely is going to be verified with the help of range of subjective brightness sensitivities showing a particular adaptation level. Let us take one of the graphical representation which in terms of your HSV your HVS is right now you are defining in terms of characters, in terms of character, in terms of study, in terms of study the characters of observation. How we are going to verify is, let you say, take the x-axis as a logarithmic of intensity value. What do you mean intensity? Is a fixed intensity. And what is the y-axis you are taking is, we are taking in terms of your subjective brightness sensitivity of a pixel. So, and based on your height, that is based on the structure of the eye, based on the structure of the eye, you are going to be understand here how that varies. So you said, right, previous information we are considering photonic. So that photo pic values, how we are comparing with the scotopic. Photopic value compare with the scotopic values in terms of human high observation. The values which you are going to be understand that how we are looking in terms of very clear. Very clear means it is in between this, how it is very clear. In between this, how it is clear. Is there any linear observation or non-linear observation? That is also we are concentrating here. Linear observation means 100% visible and 100% observation is correct. Non-linear means it may be approximate visible. So that complexity will be going to be minimized. How can you minimize that complexity? Is the range of light intensity levels to which human visual system can adapt of its order of 10 to the power of 15 from the scotopic threshold value to the glare limit. So in a photopic vision, the range is about 10 to the power of 6. Here 10 to the power of 10. So you can see how this range will be different. The range of light intensity levels to which the human visual system can adapt is enormously. We are concentrating on the order of 1010 from the scotopic threshold value in terms of experimental evidence indicates that subjective brightness is varying in terms of logarithmic function. That we can understand that the perception by the human visual system is corrected right now as intensity of value. So that intensity incident on the high will be dependent. That's what today this human visual system process, we can understand that how the human visual system process observation is depends on high structure of the high variation only. That the structure of the high is process in terms of what we call is in terms of color process. That is, the light is a form of electromagnetic energy that can be completely specified at a point in the image plane by its wavelength distribution. Not all the electromagnetic radiation is visible to the human eye. In fact, the entire visible portion of the radiation is only within the narrow wavelength band of 380 to 780 nanometers. Even still now, we are concerned mostly with the light intensity, that is, the sensation of brightness produced by the aggregate of wavelengths. However, light of many wavelengths also produce another important visual sensation called color. So that is what we are concentrating in terms of color image perception. We consider that as a more fundamental of the color image processing in aspect of visual perception of the color images. That's we are concentrating here one of the major elements of the color image processing. So we are concentrating here not only based on the light, but also different spectral distribution generally need to concentrate on concentrating on, but not necessarily have different perception of the color. But that information color is the aspect of visual radiant energy by which an observer may distinguish between different spectral compositions, different compose what we call that one is different spectral compositions, which help us to understand about color image variations. Color image variations. So coming to that, color image variations is also concentrated in terms of visual perception of the color is long slide curve represents. Long slide curve represents. That is the range of intensities to which the visual system can adapt in terms of in photophic vision alone. The range is about 106. The transition from scotopic to photophic vision is gradually over the approximate range from 0.001 to 0.1 milliambert. That is, we say -3 to -1 ml. Simply you can write ml in the long scale. As the double branches of the adaptation curve in this range as shown, which we are understood. The essential point in the interpreting the impressive dynamic range depicted is that the visual system cannot operate over such a range simultaneously. Moreover, here you are going to be concentrate in terms of accomplishes this large variation by changes in its overall sensitivity in terms of phenomenon known as brightness adaptation. The total range of distinct intensity levels it can be discriminated simultaneously is rather small when compared with the total adaptation range. So what do you mean to understand from the here is right now we are concentrating that the approximate range of information is very important. So based on that your color image observation may be different. So the perception is here observation only. So coming to the in terms of continuation where we are concentrating about this observation is varying in terms of brightness and intensity of the your particular image which you are giving into the your previously discussion about block diagram. There it's concentrating relation between brightness and intensity is not a simple function. It is in terms of image sensing and acquisition. What do you mean that image sensing and acquisition means if a sensor can be developed that is capable of detecting energy radiated by a band of the EM spectrum, we can image events in that band. Image is generated by energy of the illumination source reflected in terms of transmitted through objectives as we consider in terms of X-ray. So you can also concentrate that focus on distance object. When we say lens is relatively flattened, refractive power is minimum. Focus on near object, how it is concentrating in terms of near object is thicker, that is lens is thicker. Refractive power is maximum perception takes place by excitation of perceptors which transform radiant energy into electrical impulse that are decoded by the brain. So we understand RGB models. So brightness and intensity is concentrating on the dynamic range of light intensity to which eye can be adapted is enormous. Brightness is a logarithmic function of a light intensity in terms of which VS cannot be operated over the entire range simultaneously. So it accomplishes large variations due to brightness adaptations only. So we may understood coming to the very important slide, that is how this color stimulus we are concentrating in terms of specified by visual radiant energy of the different pixel intensity which given in terms of spectral composition. As considered, color is generally characterized by attaching names of the different stimuli. For example, white, grey, black, red, green, blue. So, color stimuli are generally more pleasing to eye than black and stimuli. So, consequently, pictures with color are widespread in TV photography and printing. So, color is also used in computer graphics to add spice to the synthesized pictures. Coloring of black and white pictures by transforming intent of intensities into colors called pseudo colors, which we are discussing in the earlier class. So has been extensively used by artistic working on in pattern recognition. In this model, we will be concerned with the questions of how to specify the color and how to reproduce it. The color specific consists of three parts. Part number 1 is color matching. Part number 2 is color differences, and part number 3 is color appearance or perceptual color. So what we can understand from the here, your colors are consists of three parts which are morely useful in terms of matching, difference, finding difference, and see the appearance. So here what we understand here, the visual perception of the color image is right now changes. Here is color image is changes in terms of your three consists of the parts. One is color matching, the second one is color differences, and third one is color appearance. So once you concentrate your perception, which we understand in terms of visual perception, which we are taking help from the HVS system. HVS system is we are prepared in terms of structure of the eye. In terms of structure of the eye. Structure of the eye. So based on this, we are going to be see the three different colors: color matching, color differences, color appearance, or perceived color. So coming to the such a visual perception of the color images, where we can use in terms of application, in terms of medical image processing, quality control system, surveillance, law enforcement, and which very important we can say artistic. So medical and artistic effects is depends upon the human eye observation only. So based on it, we find out the perfect color image processing. So you can understand this visual perception of the color image topic, which we are going to be understand in terms of number 1. Okay, number 1 is how that HVS system can be defined. Number 2, the structure of eye, how it is defined. So depends upon this application may be concentrated. Okay. So with this, I wanted to refer the some of the list of references which help us for further information which you are connecting in terms of concentrating for the further improvement of the knowledge, are going to be referred with these two textbooks only. So with this, I hope you are all understand about our lecture, which we are concentrating on visual perception of color image, how it is connected with the human eye observation. I'm very thankful for you all for concentrating on this complete video for sake of understanding as a topic from our syllabus. Thank you. Thank you so much.
Like, share and subscribe. Hit the bell icon for more updates.